It's sometimes the case that when we're looking at animal behavior, we're using them as models for other species, particularly humans, meaning we hope to successfully generalize from one form to another. But there are other dimensions of generalization that one has to be careful about sometimes. And we're going to look at 6 of these dimensions. They are not necessarily mutually exclusive, I think, as you'll be able to figure out. So let's look at the first one. Generalizing across species. Well, that's pretty much what I just mentioned, especially if you're going to study a non-human organism and try to learn something about it and then generalize to humans. But you do have to be careful. For example, can you necessarily generalize from a rat to, say, a guinea pig to a primate, a non-human primate, from a non-human primate necessarily generalize to humans? Well, you know what? Many times you can jump across all of those loops and do that, but not necessarily always. So again, this has to be done with some degree of caution. Look at bears, for example. Can you generalize from bears to humans? I mean, we're both mammals. Let's look at one form of behavior, hibernation, hibernating bears. It turns out that after about 130 days of hibernation, a bear will lose only 23% of its muscle mass. Now that may seem like a lot to you, but remember it's been immobilized for a long period of time, for 130 days. What about a human that for some reason has been immobilized for a comparable period of time? Perhaps there's been some kind of an injury and the person is going to be in a cast or in a wheelchair, some form of immobilization for about 130 days. Well, it turns out that not only for humans, but also for rats, that after 130 days of immobilization, there's a 90% amount of muscle loss. That is huge. And some of the reasons for this are because of just simple atrophy of skeletal muscles, loss of muscle tone, things like that. And we don't see that happening in bears. So in this case, there is not a lot of basis for generalization from bears to humans with respect to muscle loss following immobilization. Here's another dimension of generalization, from domestic animals back to the wild form, or for that matter, vice versa. So, looking at cats and dogs, the 2 most common domestic pets that we have, they're both from wild progenitors. Can you necessarily generalize from one dimension to the other, from either the wild progenitor to the pet, or from the pet back to the behavior of the wild progenitor? In some cases you can, and in some cases you can't. But you do have to be careful. For example, it's recently been discovered via an intensive DNA analysis that cats, relative to dogs, are really only semi-domesticated. Dogs are much more domesticated. They've been domesticated a much longer period of time than cats have been. So cats are more closely related to the wild form from which they've been domesticated than dogs are from wolves. What about across contexts, our third dimension of generalization? Can you necessarily generalize from the wild to, say, an outdoor aviary? We're looking, of course, at birds here. To an indoor cage? Well, again, sometimes yes, sometimes no. But let's look at this. Now, we're not going to be looking at birds. We're going to be looking at another species. But it drives this point home. So we're going to be looking at circadian rhythms, day-night activity cycles. It's regulated by a brain area called the suprachiasmatic nucleus. It all has to do with the amount of available light in the environment and how the suprachiasmatic nucleus outputs things involving physiology and behavior through all kinds of other pathways. I mean, we do have circadian rhythms. This is the typical human circadian rhythm cycle from various times of day. If you want to study this a little bit more carefully, you can pause. So basically, like I said, we're dealing with light. Whenever there's light, there's more activity. When there's no light, there's less activity. Not in all animals. I mean, there are animals that are nocturnal, and they're more active at night than they are during the daytime. But we, of course, are more active when there are light conditions rather than nighttime conditions. So we're going to look at Drosophila because they are more like us, the fruit fly. So it turns out that there is a context effect with Drosophila and circadian rhythms across two different contexts, the lab versus the field. Also, temperature is involved here along with light because, of course, there's going to be variations in temperature in the field condition. So there's 4 proteins that control gene expression that are affecting Drosophila circadian rhythms. They're named clock, cycle, period, and timeless. And a lack of any of those will affect circadian rhythms. But for our purposes here, we're only going to look at the clock protein. So we have clock Drosophila and clockless Drosophila. In other words, Drosophila that have been manipulated such that they lack the clock protein. Okay, so here are the data. It looks complicated, but it's actually not that bad. So across the top, we have normal fruit flies. Across the bottom, we have our clockless fruit flies. We have 2 conditions, the lab and outdoors, the field. And now in the lab, of course, light is controlled by light bulbs. So we have the lights off, the lights on, the lights go back off. And the same down here, of course. And in the field, we have nighttime. We have the sun comes up, and then the sun comes down, goes down, back to nighttime conditions. In other words, we have dawn and dusk and daylight. Okay, we're looking at 2 different things here. Temperature, that's the red line. But in terms of behavior, the blue line is what's really important. The activity cycles, the circadian rhythms. Okay, let's see what's happening. So notice in the laboratory, there are anticipatory periods in which activity starts to gradually increase right before the light goes on and right before the light goes off. Gradual increase, okay? Notice that we see the same thing happening outdoors right before the sun comes up, gradual increase, right before the sun goes down, gradual increase. What about the clockless fruit flies? Well, notice in the field, they too are showing this anticipatory rise in motor activity. But in the lab, they do not. They just suddenly start to increase their activity the moment the lights go on, and once again, the moment the lights go off. And then, of course, they decrease. So there's no anticipatory activity cycle for clockless fruit flies in the lab. Interestingly, there is an anticipatory period outdoors. So there's a context difference there. Notice also, and here's where temperature plays a role, that outdoors there is an afternoon peak for both the normal fruit flies and the clockless fruit flies. And that's because the day is getting warmer in the middle of the day. But notice that in the laboratory, because temperature conditions are held constant throughout the day, there is no temperature peak in either the normal fruit flies or the clockless fruit flies. So again, our laboratory flies, in this time both the normals and the clockless, are behaving differently from those that are outdoors. So to summarize here, clockless mutants show no anticipatory morning and afternoon activity in the lab, but they do show it outdoors. Temperature, a key factor outdoors. Now it turns out, and I didn't show these data, that when the outdoor temperature variation is simulated in the lab, so midday you make it a little bit warmer, then you get that third peak, the one in the middle of the day in the lab. So you do have to manipulate the temperature in order to do that. So, just a precaution, when one is doing lab studies, it's important to try to incorporate as many elements of the natural environment into the laboratory situation as is possible. Now, it's never going to be possible to incorporate everything, obviously, but taking that into consideration also should give some caution when trying to necessarily make blatant generalizations from one context to another, because there may be no justification for doing so, or there might be. What about behaviors? Can you generalize from one type of behavior to another? If you're doing a learning study, an operant conditioning study, is key pecking the same as bar pressing for a reward? Rats bar press, pigeons key peck. Now, of course, here is where we're sort of crossing lines of generalization, because we're also using different species and slightly different contexts, but not really. They're both Skinner boxes, just designed a little bit differently. But I'm focusing here mostly on the actual behavior. Is one behavior for a reward equivalent to the other behavior for a reward? And I have no answer for that question, by the way. But look at this. Another type of thing that's used in the lab a lot is a Y maze. So there's a food reward at one end of the Y, and the other end of the Y has no food reward. The animal should learn to always go wherever there's a food reward. Okay, so they're learning where the reward is. Very simple task. But you know what? Things are a bit different in the wild. So again, we're crossing our dimensions here because we're going from a laboratory situation to a wild situation. But again, here I am focusing mostly on the behavior per se, running down a Y maze versus, for example, if you're an acorn woodpecker and you're caching acorns, you got to remember where you've put the food. In a Y maze, it's going to be at one end of the maze or the other end of the maze, one extension of the Y or the other extension of the Y. It's pretty easy. In the wild, it gets more complex. But acorn woodpeckers drill these holes and cache acorns in them. That's why they're called acorn woodpeckers. And they remember their cache sites. They remember just about every hole that they've dug. So they'll know exactly where to go to retrieve their acorns. And they're not the only caching species. There are some others that are quite amazing. For example, if we look at Clark's nutcracker, I mean, these guys are really amazing. Check this out.
It depends on pine nuts, but these nutritious nuts are in season for just 3 weeks in September. The bird flies as far as 12 miles between the pine trees and suitable sites to store the seeds. The nutcracker buries the nuts for later. It drives them one at a time into the hard soil, and the spot is sometimes marked with a stone. In the short time of plenty, the nutcracker has to learn the locations of 30,000 nuts it buries at sites scattered over hundreds of square miles of the Grand Canyon. Its landmarks aren't remembered like photographs. Snow transforms the landscape. But this doesn't confuse the bird. It finds 90% of the 30,000 pine seeds it buried. It's one of the greatest feats of memory in the world.
It's pretty amazing. Even when the ground is covered with snow, they're still remembering 90% over a very large area that they have to cover where they've cached their nuts. So how do they do that? Well, pretty much the same way that we would. We're not necessarily that good at it, But like us, they have a hippocampus in their brain. And we're looking at just a picture of the hippocampus of a black-capped chickadee in this particular photograph. The hippocampus is hugely involved in learning and memory across all species. It's involved in other things as well, but we're focusing on memory of cache sites here. So let's look at three closely related species, the black-capped chickadee, the Mexican chickadee, and the bridled titmouse, maybe a little bit less related for that bridled titmouse. Okay, so what we're looking at here are the number of caches typical of each of these species and the volume of the hippocampus. Is there a relationship there or not? So the black-capped chickadee has the most number of caches. They cache more than the Mexican chickadee or the bridled titmouse. What about the volume of the hippocampus? Well, it turns out that the black-capped chickadee has a larger hippocampus than either of those other two species, where it's about comparable. Even though the bridled titmouse makes fewer caches than the Mexican chickadee, the volume of the hippocampus turns out to be the same in those two, but it's significantly greater in the black-capped chickadee that makes significantly more caches. So they have a good memory for where they've stored things. Here's some closely related warblers. Okay, so this is just another example. We have the garden warbler is a long-distance migrant. It has a larger hippocampus. So when it migrates, it just covers hundreds and hundreds of miles. Here is a closely related species, the Sardinian warbler. It doesn't migrate at all, whereas the garden warbler does. So it pretty much stays where it is year-round. It has a smaller hippocampus. Now, with that in mind, is there a relationship between where these guys remember their cache sites? Well, in this case, it was done in a laboratory, so it's a highly controlled study. And it turned out that for the garden warbler, they can remember where things were stored for 12 months. Food, those things. Whereas for the Sardinian warbler, in the lab tests, only 2 weeks. So yes, the larger hippocampus is very, very important for not only remembering where you're going in terms of migration, but also in the laboratory for where food happens to be stored. Okay, so let's get back to our dimensions of generalization, having looked at those examples on caching and hippocampuses. Rewards. Would you work as hard for a piece of bread as you would for a brownie? I wouldn't. But, different people have different tastes. In other words, can you generalize from one reward to the next? Because if, for example, you are doing a learning study, there may be differences in what you see based on what you're rewarding the organism with. Here's a good case in point with capuchin monkeys.
So what we did is we put two capuchin monkeys side by side. Again, these animals, they live in a group. They know each other. We take them out of the group, put them in a test chamber. And there's a very simple task that they need to do. And if you give both of them cucumber for the task, the two monkeys side by side, they're perfectly willing to do this 25 times in a row. So cucumber, even though it's really only water in my opinion, but cucumber is perfectly fine for them. Now, if you give the partner grapes, the food preferences of my capuchin monkeys correspond exactly with the prices in the supermarket. And so if you give them grapes, that's a far better food, then you create inequity between them. So that's the experiment we did. Recently, we videotaped it with new monkeys who had never done the task, thinking that maybe they would have a stronger reaction, and that turned out to be right. The one on the left is the monkey who gets cucumber. The one on the right is the one who gets grapes. The one who gets cucumber, note that the first piece of cucumber is perfectly fine. The first piece she eats. Then she sees the other one getting grape, and you will see what happens. So she gives a rock to us, that's the task. And we give her a piece of cucumber, and she eats it. The other one needs to give a rock to us. And that's what she does. And she gets a grape. And she eats it. The other one sees that. She gives a rock to us now, gets again cucumber. She tests the rock now against the wall. She needs to give it to us. And she gets cucumber again.
So obviously, if you were going to do some kind of a learning or conditioning study with these guys, I think grapes would be a better reward than cucumber. Can you generalize across sensory modalities? As we see in this cartoon, dogs have issues with seeing color, at least the way humans do. So no, you can't necessarily always generalize across sensory modalities, because it turns out that for humans that are not colorblind, at least, we see a lot of colors. However, dogs, on the other hand, don't see all those colors. They are, in fact, what we would call red-green colorblind. They don't see reds, they don't see greens. So, if you're using a dog in a study in a lab or something, and you're trying to do a color discrimination experiment, you would not want to use red versus green. They don't see those colors. They might see shades of gray, but that's about all they would do. No, you should capitalize on what they are capable of seeing. Okay, so those were our 6 dimensions of generalization. It's very easy, and we've seen this a number of times, all too many times, where people get very sloppy and they're not careful about the kinds of generalizations they make. They make sweeping, sloppy generalizations. We see it in the popular press, the media, all the time. Scientists try to be much more careful. Not necessarily all scientists, though. Especially some of the popular books that have come out of ethology. So again, I emphasize these tend to be more popular books, not things you find in scientific journals. But, you'll see some problems there from time to time as well, and not necessarily only in ethology. Konrad Lorenz wrote a popular book back in the 1960s called On Aggression. And he was saying in this book, basically, the premise of the book is that that non-human organisms are aggressive, and therefore so are we. We are destined to be a very aggressive species. Just the problem with that is that there are different times when you'll see aggressive behaviors in non-human organisms than when you'll see them in humans. I mean, humans can be very aggressive for no particular good reason at all, other than to be mean to somebody, whereas that's not generally what you're going to see in non-human organisms. Typically, it's related to territoriality, getting a mate, keeping other males away from your mate, or whatever. But it's not necessarily just going to happen like it might just happen all of a sudden in us. So not all forms of aggression are equal. So it can be dangerous to generalize across species with respect to these kinds of things. But again, that was the entire premise of this book, which actually was a bestseller. I think people bought more into it than they probably should have. Lorenz wrote another popular book, Civilized Man's 8 Deadly Sins. I think this was the last book he wrote, certainly the last popular book that he wrote, because by this time he'd already won the Nobel Prize in ethology. So these are the contents, and if we look at what these sins are, they're the ones you see here: overpopulation, devastation of the environment, man's race against himself, entropy of feeling, meaning a lack of feeling, genetic decay, the break with tradition, indoctrinability, we are easily indoctrinated, and nuclear weapons. Okay, those are our sins. Well, let's just look at this one, genetic decay, because it involves domestication, one of our dimensions of generalization. Look at this quote from Lorenz. The immensely high reproduction rate in the moral imbecile has long been established. This phenomenon leads everywhere to the fact that socially inferior human material is enabled to penetrate and annihilate the healthy nation. The selection for toughness, heroism, social utility must be accomplished by some human institution if mankind is not to be ruined by domestication-induced degeneracy. The racial idea, as the basis of our state, has already accomplished much in this respect, with the extermination of elements of the population loaded with dregs. Oh my gosh. So, Lorenz has written many articles where he's expressed his views of domestic animals as being behaviorally degenerate, where he talks about how they've lost behavior patterns that are found in the wild form. And we'll look at this in more detail when we deal with domestication later in the course. And the point I'm going to make then, as I'm going to make right now, it's a faulty view. And then down here, it's like even worse, extermination of elements of the population loaded with dregs. I mean, I think he's talking about genocide, which is horrible. So, it's very difficult to deal with some of these semi-popular books. But nonetheless, I just wanted to point out these dimensions of generalization. We need to exert caution sometimes, many times, but it doesn't mean we can never do it. It means we just do so carefully.